All right, gang. Welcome to the Unit 2 review for Principles of Biomedical Science. Talk about the test of treating the patient, right? So first unit was all about coroner reports and solving a kind of a mystery. This one also is a mystery, but we're treating the patient, right? So first thing we're going to do is take vital signs, right? So vital signs is the thing that we take at the doctor's office or sometimes we can take these in the field if you're a paramedic, but what we're going to look for are 5 major things. Number one, we're going to look for the heart rate. So heart rate, remember, is the beats per minute. That's basically all we're taking here, right? So this, we talked about how you could take this both with a device that can capture that information for you, or you could do this manually. Usually you're going to take this from a radial pulse, which is the one that is along your wrist area, right? In this area right here. If you cannot capture that, remember that you never want to use your thumb when you're taking a heart rate. You also can take it here in the brachial artery. You could also take it along the femoral artery in the groin region if neither one of those first two. You can take it up here in the carotid artery as well. But generally, we take it at the radial pulse. It's pretty easy to capture, and you could easily Google what's the common resting heart rate for a normal situation would be, right? Blood pressure is slightly different. So this is the force of the blood against artery walls. And we capture this in 2 different levels. So upon a contraction and in relaxation. And so we generally get something like 120/80, but that can vary as well, right? Based on age and gender, all that kind of stuff kind of plays a part in all of the what would be considered normal or healthy for the individual. But this is called systole, and this is diastole, right? Oxygen saturation is the amount or the concentration of oxygen in the blood. And we're going to be looking for numbers that are close to 100%, right? So we're looking at 94, 95, 96% and above. Below that, we'd say that they're having some situations that would warrant some sort of intervention. Respiration rate is the amount of breathing, breaths per cycle, breaths per minute, right? All those types of things can be looked up. And then temperature, we're looking for core temperatures, and we want somewhere around 98.6, right? And that could fluctuate a little bit. But these are considered vital signs. All of these things, make sure you realize that these can all be influenced by age. They could be influenced by different types of diseases, both pathogenic ones, right? So ones caused by bacteria and viruses, fungi, but also genetic diseases, which we talked about near the end of this unit. This can also be influenced by male, female, right? All these things. And then, in addition to that, things like stress or food, right? What you're eating, right? The obesity, the thinness, or all that kind of thing. Thinness, the amount of weight the person has can all influence your vital signs. Looking, right? This is the next level. When your patient comes in, you need to look at the patient. So looking at their skin to see if there's any sorts of abnormal looking components to the skin, like is there rashes? Are there abnormal colorations? And when we were saying this, we were focusing mostly on pockets of color that could possibly lead to things like cancer, right? So we're looking for melanomas and things of that nature. Carcinomas is the generic form of cancer in the Greek Latin. I know carcinoma sounds very different for you other than saying cancer, but we're focusing on cancer being this bigger thing that we're going to discuss on another slide here. And carcinoma is just saying that it's a cancerous body, right? Those cancerous bodies can be different based on where they crop up. We're focusing here on skin as skin cancers, right? You also look in the mouth to make sure that there's not abnormal bumps or enlargement of any of the glands, specifically focusing on tonsils. We also want to look for abnormal discharge. All right, we shouldn't see weird coloration like white spots or anything like that. Right, we don't want anything like that in the back of the throat. Green, things of that nature that might show signs of infection. Nose, same types of thing, right? We're looking for clear mucus. And also no inflammation. Right, and when we go into the case study, you're going to be looking at images of that so that you can designate whether or not there might possibly be an infection there. Same thing with the throat, right? Mouth and throat connected together. We're looking for bumps, enlargements, things like that. Ears. We should see a normal amount of ear wax. So there should be wax in there that is like a yellowish color. But more importantly, with an otoscope, you should be able to look all the way down to the eardrum or the tympanic membrane that's back there. And if that eardrum is fairly clear, you should be able to see the auditory ossicles. So that's the malleus, the incus, and the stape, right? So those three bones on the inner ear, you should be able to see those as well if there's no infection in that ear. If all that stuff is looking good, you also need to take, when the patient comes in, a quick listen, right? So listening to the heart, you should be hearing a nice rhythm. So good rhythm to the heartbeat, specifically listening for a good solid lub dub, strong lub dub. We don't want to hear any hissing noise or crackling. Those are bad, right? That means that the alveoli or the bronchioles have mucus that's in there that's causing air to not move through properly, and it causes pockets, and then that force of those pockets pushes and it causes those abnormal noises like hissing or a crackling or popping noise. Those are bad, right? And we should hissing, crackling, that also kind of connects with lungs. Specifically with the heart, the hissing is going to be connected to valves not closing properly. Here, lungs, I was misspeaking, I apologize. The lungs, we're talking about nice in and out whoosh kinds of noise. Whoosh. We don't want to hear no on these for hissing and crackling, right? No hissing, no crackling, either from the heart or from the lungs. Mostly the lungs is where you're going to hear crackling if things are in a bad situation. Now with HIPAA, we need to understand the basics of what HIPAA is and what it all means from that perspective. So this is the health insurance. Health insurance portability and accountability sorry for writing. My handwriting is really bad today. Act. Okay? So what this is saying is that your health records are yours. Right? That's it. They're not anybody else's business. And if they get shared without your knowledge, that is a violation, and you have legal course that you can take to fix that. Okay. We then spun a little bit into immunity and how your body fights diseases. So there's some key things here that students goof up. Number one, antigens. Remember, these are like the cell flags that we talked about. So if we had a cell, we had something sticking off of it that had a picture of you, this is your antigen, right? So this is a good, healthy cell. You'll have a nice, healthy flag that's out there. Antibodies are your weapons against invaders. And the cells are the things that are going to respond. So we have different types of what are called white blood cells that are going to be the main workers for the immune system. Remember that we talked about macrophages. And we talked about helper T cells. And I'll talk through these on the next slide. Helper T cells, killer T cells, and B cells. These are the main ones that you need, at least for immunity. And then there's this lines of defense situation. So you have that first line of defense, which is basically your skin and mucus, right? Stops a lot of stuff from getting inside your body. But if that virus or bacteria pass the first line of defense, we get to the second line, which is not very specific, so it's not specific. And it attacks pretty much anything, and that's the macrophages involved with it. And then if we get past that, we get into the third line. That is where things get specific, and that's where I'm going to kind of focus on this next slide here so that you have understanding. So the specific line of defense, if we had a pathogen that came in, all right, so here's our little bacteria. And that bacteria comes in, your cells have the ability to gobble it up, the white blood cell. So here's my white blood cell, surrounds it, engulfs it. His eyes are not so good. There you go. There, that's a little better eye. Engulfs it, and then what happens is this white blood cell, this macrophage, will present part of that bacteria on itself, on that antigen, right? So now we know what the bad guy looks like. We then have the helper T cell, which is the smart one, if you remember those videos that we watched. It's the smart one that looks at it and goes, yes, I see, and it calls in the rest of the army. So it calls in the killer T cells, and it calls in the B cells, so that we can get some specific defense against that invader. So the killer T cells will go after your cells that are infected and destroy them so that they don't allow the spread to continue. Your infected cells, it kills those, right? So let's say you had a regular body cell, the virus or bacteria got inside it, started making this cell really bad. You could kill this cell so that it doesn't spread its badness onto other cells, right? Or spread it back into the bloodstream. And then the B cells are the ones that we hear about the most because they're the ones that produce those antibodies. Remember, those antibodies, they're kind of Y-shaped a lot of times, are really specific. And that's why they take a little while to produce, because they will specifically be able to attach to these bacteria or viruses and stop their spread within your body. Okay, that's really important for you to understand. And it does connect, as we saw in the documentary, to cancer, because if we can teach your body to produce, or teach your body that the invader of cancer, which is your own cells, is bad, we can keep your system on for killing it. Because remember, cancer kind of tricks your killer T cells to say, no, you don't have to kill me. So we can use antibodies to stop that communication and allow your cells to continue to kill the cancer. So how does it connect to mitosis? Remember, mitosis is just cellular division, right? So this is the IPMAT, right? Interphase, prophase, metaphase, anaphase, telophase. And this is basically just cellular division. And the whole goal here, ladies and gentlemen, is one cell turning into two cells that are exactly the same. They're clones of each other, right? They're exact copies of the original cell. We are trying to do this to repair damage. We're trying to do this to grow, right? Those are the main things that your body's doing this cellular division. Your hair needs to keep growing. Your nails need to keep growing. You need to keep growing new skin because you constantly damage it. That's mitosis, right? Your cell will sit in these different phases for different amounts of time. Remember that we talked about how interphase is the big one. That's your G1, your S, and your G2, whereas mitosis is just that little wedge. So we're only doing mitosis when we want to do cellular division to create new cells. So G1, it's doing its normal thing, doing its celly cell stuff. S, it's going to replicate the DNA. So if we had the DNA all packed up into a chromosome and we could actually see it in G1, it would look like that. In S, it would finally get its X shape, and then in G2, it stays that way. Because when we go through mitosis, it ends up back like this, right? So we end up having these two cells. One will continue back around again, right? But we end up having two cells out of that division. Now, where cancer comes in is that's when this is uncontrolled mitotic division. So your cells should stop, but they're not stopping, and they're continuing to divide. That's nefarious. That's bad. That's really, really bad, because it could take nutrients from the healthy cells, and it could take up space, and we don't want that to happen, right? So cancer is just uncontrolled mitotic division or uncontrolled cell growth, right? Tumors are generally the clumping of that cancerous situation that's there, okay? Speaking of chromosomes, you have to understand what chromosomes are. So a reminder that if we have DNA, which we're going to talk a little bit more near the end of this, DNA, we take that DNA, remember you've got 6 to 7 feet of it per cell, nice long stringy stuff, and we take it and we wind it around some balls of protein called histones, right? And then we let those balls of protein also wind around each other. I'm trying to do this very fast. It's not showing up very well. And we keep winding the winds together, right? So wind the wind. Eventually, you'll end up getting a structure that you're familiar with that is this structure, right? So that's the chromosome. So this is a chromosome. But keep in mind that also this is a chromosome. So both of these are chromosomes, but this chromosome on the left is a chromosome with chromatids. Because chromatid just means it has a replicated copy attached to that B, which is the centromere. That's the thing holding the top half and bottom half together. It also is the way for the two chromatids to attach, okay? So that centromere right at the middle, we have these sections within the chromosome like A is showing, which maybe you would call a gene, which is just a section of the chromosome that codes for maybe a specific protein or a section of a protein. And so if this chromosome here, which I've written all over, was replicated in that S phase, and the left side over here, it said big A. Well, remember the right-hand side is a duplicate, so that would also be big A. But remember, you have two copies of a chromosome, one for mom and one for dad, so you could have another chromosome that replicated at the same time, and it could be little a and little a. So what is your genotype? It's not big A, little a, it's just big A, little a. Because remember, your chromosomes aren't like this unless they want to do mitosis. If we rewind, right, your normal situation is up here in the interphase, not down there in the M, right? We only do the mitosis when we want to divide, okay? So then that took us to genetics. So in genetics, we're talking about the inheritance of those genes and how that plays a part in producing your phenotype, how you end up looking. So we had just the basic stuff from your previous science-y class that you took that covered some genetics, or it's dominant recessive, where you had big R, which can mask the little r, and you get red, right, which would be heterozygous there. Big R, which would also be red, and then you might have little r, which is white. So that would be just a normal dominant recessive situation, which is the ones that generally for you are going to show up in the Punnett squares and the pedigrees and things of that nature. Whereas incomplete dominance, it's kind of like a blending situation. So we would have something where if you had both alleles, both kinds of alleles, you end up getting a blend between the two. So if you had a red flower crossed with a white flower in our examples of flowers here, you would end up getting a pink. That would be an incomplete dominance sort of situation. Whereas codominance, we see both of the both of the homozygotes traits in the heterozygote. What do I mean by saying that? I mean that if you crossed a red with a white, you would end up getting a flower that is red with white spots. Right? You're seeing both of the homozygous individuals in the heterozygote individual. If that makes sense, right? The phenotypes there. Polygenic was when we said that you take gene number 1 and gene number 2 and number 3 and number 4, and they all work together to produce this one specific thing, which is hair color, something like that. Right? And this is more common than a dominant recessive situation for a trait. Many genes work together to produce the phenotypes that make us us, or even a dolphin, or a cardinal, right? Pleiotropy is kind of the reverse. So here we have one gene causing all of these different effects. So this one gene will affect how your heart works, and this one gene will affect how your skin develops, and this one gene will affect your blood, etc., right? That's pleiotropy, which is, like I said, kind of the opposite in idea from polygenic. Okay, so then we looked at Punnett squares, right? So Punnett squares, you have to understand how to do these both from an autosome situation and a sex-linked situation. So remember, autosomes, those are the chromosome pairs 1 through 22, and the sex-linked ones means that we're talking about 23. So if you remember, we had a karyotype. I'm not going to draw out an entire human karyotype with 23 pairs, but remember it was numbered something like that. And then down here at 23, you might have had a big one and a small one. That would have been a male, or 2 big ones that would have been a female. So when we're talking about autosomes, we're saying that it's pretty much the same male versus female. And in sex-linked, we're saying that there is slight difference, male versus female. So in an autosome, Punnett square, all you're doing is like we've normally done. So you have, for example, big R, little r crossed with big R, little r. You just drag and drop once you put them across the top. So mom goes on the top, maybe, and dad goes on the side, and then you combine them together. So down and left, or down and from the left, and then down and from the left, and down and from the left, and down and from, right? And so you just put them in like we normally do. But what gets funky will be if we do a 3-trait cross, right? So if we did big R, big T, big Q, little q, crossed with big R, big T, big Q, little q, you have to do 3 Punnett squares. You're going to do Punnett squares for the R's, right? Oops, it's 2 big R's. You'd have to do Punnett squares for the T's, and then you have to do a Punnett square for the Q's. And then, once you figure that part out, then you take each one of these. So if we were doing like, hey, what's the possibility of being red and tall and big Q being quick? Then you would take, well, in this one, all of them turn out red. So that's 4 out of 4. And in this one, tall is 3 out of 4. And then quick is 3 out of 4. So you just multiply all of it together, and that would be, what's the possibility of getting a red, tall, quick child, right? So that's that. But then in a sex-linked, it's slightly different because the X and Y are, well, different, right? They have more, the X has more genes on it than the Y does. And so generally, if you talk about like red-green color blindness or hemophilia or Duchenne muscular dystrophy, the X chromosome is the one that holds it. So let's say you had a female that had heterozygosness for hemophilia, and then a dad that didn't have hemophilia, making an X, and they have a child, right? When you do the Punnett square, you have to do it with the X's and Y's, but the Y doesn't have anything on it. So when you put it into the Punnett square, what's the chances of them having a daughter that would have hemophilia? Well, when you cross this in, I don't know how, that's pretty small. Answer is 0, right? Their daughters cannot have hemophilia. But what are the odds that they'll have a boy that has hemophilia? Well, that is right here. So that's 1 out of the 2, so that's 50%. So that's a slight difference, right, in how you're doing the Punnett square. So do make sure you're on board with that situation. All right, last thing before we get into kind of the pictures here is CRISPR. So realize that this is a technique we can use to do gene editing. How does it work? Well, there's this enzyme called Cas9. I'm just going to draw a blobby blob. So this is Cas9. And Cas9, if you give it a piece of DNA or a piece of RNA, that we will call it a template, for them to use to look for the gene that you want. So here's the DNA. I did a horrible job drawing the DNA there. And we know a specific gene like sickle cell, what it quote unquote looks like, right, in the nitrogen base sense. Then we can make a template that matches that. So this is sickle cell. And then we could throw Cas9 with this little template into your DNA. It finds where it matches up, bing, 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 right here, and it's able to cut it on both sides and get this piece out. If you do that and you inject this Cas9 with its template and a section of correct gene for hemoglobin, because remember, sickle cell is an effect on the hemoglobin protein, then you can essentially fix this person. So they are born with sickle cell, and now through CRISPR, they don't have sickle cell anymore, right? So they've been cured of that sickle cell without a pill, okay? All right, so DNA replication. Picture-wise, you're going to have to know what it kind of looks like, but you're also going to have to know what is going on here. So the whole purpose of DNA replication is literally to make a new piece of DNA. So to turn one copy of DNA into two copies, and they should be the same. So it requires a bunch of enzymes. You need to generically know what they do. So W here / D, right? Actually, W is called topoisomerase, but I digress. D in this picture / W is going to be called helicase. So helicase unwinds our DNA for us. These are called single-stranded binding proteins. We didn't really talk about them too much in this class, but they're going to keep that fork open so that we can actually get in at the nitrogen bases so that we can replicate them and lay the new pieces down. Z in this picture is RNA primase, because if you remember, or primase, it lays the RNA primer so that DNA polymerase can add its pieces, because DNA polymerase needs to actually touch another nitrogen base on the green side, or the green side on this here, and be able to go in a 5 to 3 direction. So B is just a new nucleotide, right? In this situation, that would be RNA based. So that's what you're kind of seeing here. The RNA polymerase is going to add that RNA primer in. And then the X's are going to be your DNA polymerase. So they add the new pieces of DNA that would be a complement to the other side. And then, obviously, you should probably know that. So it's A with T and G with C. Don't forget that, okay? And then once you have that accomplished, there's going to be sections like the lagging strand in the picture here that has fragments, and we got to glue those fragments together. So then you have Y here that is ligase that will be able to do the covalent bonding required to glue those Okazaki fragments, or for your sake, just the fragments together, okay? So don't get this confused with, so this is us replicating the DNA. If we zoom back out, right, from the perspective of our cells, we're doing that in the S phase. For what purpose? To produce new cells. But then also we have to be able to understand that that's just replicating new DNA. It's not actually making you. You is protein synthesis. Because remember, protein synthesis is taking DNA, turning it into mRNA, and then turning it into an amino acid chain. And that amino acid chain is your phenotype, right? How you look, the way you sometimes even you behave. That's all connected to your DNA turning into an amino acid chain. So the DNA to mRNA section, this is transcription, just review. And that's going to happen in the nucleus. And we use RNA polymerase to do it. Whereas this picture right here, this is showing you translation, which is a little bit more complicated from your perspective. So translation, we're going to use this ribosome And that ribosome is made out of protein and rRNA. Here's the mRNA coming through in the picture. And then we have like these spots where another type of RNA can come in and start doing its job of assembling or putting these amino acids together. So this jagged blue piece here, that's a tRNA, and it's bringing in an amino acid. So what you see in the picture here is we have tRNAs that drop in. So here it's dropping in here. It's going to then take its amino acid and bind it to the chain of amino acids that might already be there. And then what's going to happen is this one will move over to this spot without the actual amino acid attached. This one right here slides into its spot and the chain is now bigger. So now a new one can come in on this side and it can continue the process. And the whole goal, once again, is to make that amino acid chain, which then can fold into all kinds of crazy shapes and become the different proteins that help run your body. Because remember, we told you that proteins pretty much do everything. They are the protective features of your skin, hair, and nails, and they're also enzymes that help run reactions, and they're transport molecules in your red blood cells, and they're also communicatory and insulin for your cells to be able to take in glucose. So there's a lot of different things your protein does. All right, another picture you'll be able to see is something like this, and this is connected with mutation detection. So remember, this is a karyotype, and so what you need to be able to discern is that there should be 2 chromosomes per number, and if that's off, that's a problem. And you can see here, this one is off, right? Because if you look at the 23rd pair way down here, we don't have just 2, we actually have 3. So they have 2 X chromosomes and a Y chromosome. So this is a situation that is a mutation. It's called Klinefelter syndrome, which we gathered from the gizmos karyotyping gizmo that we did. Okay. The other thing that you can do is this situation, a gel electrophoresis. So in this, you're looking specifically for alterations in the actual DNA. So I'm going to draw a little piece of DNA here. So remember that if we have a piece of DNA, we have 2 different versions of that gene. We're going to call them big A and little a. They possibly could get cut in different spots by a restriction enzyme. So, for example, if this was big A as a genotype, remember that that's the same exact information. So they are going to be the exact same spot, right? Boom, bloom. And they move through the gel because remember, small pieces move towards the positive faster and the big pieces stay closer to the negative. They move slower. So this piece, this A piece, is going to be a lot bigger. This capital A piece is going to be a lot bigger than maybe the little a. So what happens here is the restriction site never gets encountered. So it's a nice big piece. We're just going to call this piece X. So that's where X shows up on the gel. But then let's say A gets thrown in there. And A, you end up getting something like this where the restriction enzyme cuts here because there's a slight alteration. And now you get pieces Y and Z out of it. So you get a piece Z that's a little smaller. So that's probably this one down here, right? And then we have Y that's big, but it's not as big as X. So that's this piece right here. And so you have to be able to take that information and then be able to figure out what the genotypes are of all these other different individuals. So the first one, where we marked big A, it's the same allele, so they have the same band, right? They're the same size. Because remember, the gel electrophoresis is only separating based on size. But then in the second one, right here, we actually have 3 bands. So we have an X band, and we have a Y, and we have a Z. So that means that they are big A, little a. But then what about this one? Well, they only have a Y piece and a Z piece. So that means that they are little a, little a. Oh. And then once again here, we have 3 pieces. So that's big A, little a. This one matches the first one. So that's big A, big A. And then this one is also big A, little a. And you have to be able to transpose that onto something like a pedigree. So if I said little a was having sickle cell anemia, right? And they are colored in is the disease, then individual 1 of generation 3 is little a, which means these individuals, they might have a big A because they don't have sickle cell, but they had to give the little a to their son, so they both have to be heterozygous. But then you look at individual 3, well, what is his parents? Well, mom definitely has a big A, and dad definitely has a big A, but do we know if they are big A, little a, or big A, big A? Well, we don't have enough information to figure out if mom is heterozygous and dad is heterozygous, or if mom is heterozygous and dad is like that, homozygous, or if it's mom that's homozygous, right? So we would just keep all the possibilities until we get more information on the situation. And that pretty much wraps it up, gang. If you have questions, obviously reach out. That pretty much goes through your entire packet there.